Now, in this lecture, we want to talk about viruses and prions. Okay, so virus-like organisms we want to talk about along with viruses. So there will be two different set of lectures for viruses. One is this one regarding the structures and properties of virus and how they infect, how they multiply, and there will be another lecture on human viruses and some very common infections and diseases caused by the human viruses and its cure. Okay, so let's begin with this first lecture on viruses and prions. The very first thing that I want to share is the search of the virus, elusive virus. And the search began not very earlier. It's until 1884. Not until that time we discovered virus, because virus is so small that we cannot know the presence of virus with the help of even light microscope. Light microscope is not enough to give us the details about the virus. So when the light microscope was invented, many uh, organisms which we cannot see with naked eye is visible, like bacteria, protozoa, but not the viruses. Until 1884, where Louis Pasteur postulated that the rabies was caused by something which is much smaller than bacteria and which has a different way of approaching and infecting organisms and hosts at 1884. So again Louis Pasteur started that thinking, and then 1890s, uh, Ivanovsky and Bejernik showed a disease in tobacco was caused by virus, and then this term, this virus, this is tobacco mosaic virus, and the disease which is very common in tobacco is actually caused by virus, which is funded by then different industries. And the work start to begin in 1890s. You can imagine it's not that far away. So the research gradually begin from that time period, and by the time at 1950s, virology was a it's a, what I can say, multi-phase discipline. Okay, so where we have discovered a lot of different type of viruses, non-cellular particles with a definite size, shape and chemical composition. We know that they have proteins in it, they have some other chemical components in it, but they are not a true living organism, because they are not behaving the same all the time. That means once we provide them with a living cell, they behave like a living organism. Once we take away that cell, they are not behaving like a living organism. So the property and the nature of viruses depends on the presence of another living cell, which is known as the host. So whenever you find the host, the virus is living. Without the host, the virus is a non-living material. That's why you cannot even put virus in that three domain concept. Virus is totally separate. The foundations of virology, if you think about, this is a fascinating field, and you probably have heard of this term viruses, and every single layman even heard this term virus, because whenever something critical is thought about, something which is a very dangerous, which can make you sick and fall sick, example is virus. Because this term is used not only in disease for a human welfare, as well as in computer technology in this field, the computer virus. So whenever say something can destroy things, or is bad for your health of your PC, that's the virus. Non-living agents that infect all life forms, example phage versus animal virus. Animal viruses go and attack animal cells, while phages attack bacteria. That's why we call them bacteriophage. Viral cultivation differs from bacterial cultivation. Near about 1500 known viruses are there, and estimates to be near about 4 lakh type of viruses are there, and among them we only identify and know 1500 type. And the more we develop the electron microscopy, sorry, it allows us to visualize viruses with much up close, and we gather much more information regarding the different body component of a virus. Like in case of bacteriophage, this is a picture of a typical bacteriophage, which has a head, a collar, and the helical sheath, the tail pin, spikes, and all. So what are the general characteristics of virus? If you look at this pictures given here, all these three pictures are electron micrographs of different size and shape viruses. These are round shaped virus. This is also a rod shaped virus. This is like a very rope like coiled virus. So viruses are obligatory intracellular parasites. Intracellular means they love to be present inside the cell, and they take up nutrients from the host, and they can virtually make the host very sick and can kill it. They are filterable. The virus is Latin for the word poison. They contain DNA or RNA. They contain a protein code, okay, and the code is known as capsid, made up of capsomeres. Okay, they are of various shapes. These capsids are of various shapes. Some are enclosed by an envelope, some are without the envelope. Those one without the envelope are known as naked virus, those with the envelope known as enveloped virus. Some viruses have spikes, which are mostly made up with proteins, and most viruses are tissue specific. So they have this tissue specificity. That means some certain virus will infect a certain type of tissue. For example, hepatitis B infects the liver tissue. Host range is determined by the specific host attachment sites and the cellular factors that a virus has. Okay. Now viruses are of different shapes and sizes. You can see this picture, and that can help you understand how much diversified these viruses are. And you can see this E. coli seems pretty big of a cell in contrast with the size of the viruses, where you can see this is the red blood cell, 10,000 nanometer in diameter. E. coli near about 3,000 to 1,000 nanometer, in this case of a rectangular rod shaped structure. While a virus, if you think about, most of them are very very small. You can see even the smallest one, bacteriophages, near about 24 nanometer. So the E. coli 3000 by 1000, and in contrast bacteriophage 24 nanometer only. Other than poliovirus 30 nanometer, adenovirus 90 nanometer. You can see it contains much more spikes like that. There are square viruses or like rectangular viruses like vaccinia 300 by 200 by 100. And you can see whenever we show you the different size and shapes of virus, you can see it's all kind of geometrical shapes that are available. They are polyhedrons, so they have different planes and multiple planes are present, and we can see different kind of structures. You can see viroids, like this kind of rod like structure. Tobacco mosaic virus, another rod shaped structure, very long 250 nanometer by very thin 18 nanometer thin. You can see bacteriophage T4, that is 225 nanometer, and bacteriophage M13, you can see 800 into 10 nanometer only. Rabies virus 170 to 70 nanometer. So there are different type of shapes and sizes, and you can see Ebola virus is pretty big, 970 nanometer, near about E. coli's 1000 nanometer range, but it's a totally different of a structure. So, and you know, there's a huge place of viruses and virus community that we don't know about. We only know very fraction of that virus community. So maybe many other variety of viruses are available. Viruses, as I said, are polyhedrals. So why polyhedrals? Because of the different planes they have. They can be either this round shaped measles virus, herpes simplex virus, which is enveloped in this case, as you can see, and there are these are the shapes: poliovirus, parvovirus, okay, polyhedral shapes that are present always. Okay, so either icosahedral and different kind of. So these are icosahedral viruses, as you can see: herpes simplex virus, poliovirus, parvovirus. Now, poliovirus and parvovirus are icosahedral viruses without the capsule, but so they are naked viruses, and herpes simplex is enveloped viruses. Helical virus, as you can see: tobacco mosaic virus, rabies virus, measles virus are helical, and there are complex virus which does not have any fixed geometrical structure. They have multiple structures linked. For example, bacteriophage. Head is icosahedron, and there is this the rest of the region of the body. As you can see, apart from this region, small linker is the collar, and the rest of the contractile body portion is of like a tobacco mosaic viral helical pattern. So they are composed of multiple geometrical shapes. Now the viral structure can be divided into two different types, the covering and the central core. Okay, so the covering can be of two different types. Capsid must be present in a virus, that is the protein coat inside of which the genetic material is present. Now there may or may not be envelope. Okay, if envelope is present, enveloped virus. If envelope is absent, then naked virus. Central core can consist of DNA or RNA, that is nucleic acid molecule, or they may also contain, along with that, they also contain enzymes. Not found in all viruses, but in very few cases there are enzymes present. But most of the time they only have DNA or RNA in the core. Okay, so viruses bear no resemblance to cells. They don't have any protein synthesizing machinery at all. That's why they are totally different compared to any living cell. They don't have that idea of protein synthesis, so they are not involved in the physiological properties of a cell. They don't follow those rules. So how they survive and how they replicate, how they make their more copies? They can only make more copies when they attack their host. Once they are attached with their host, they hijack the protein synthesis machinery of their host, and then they utilizes the host's enzymes for the protein synthesis. Okay, general structure. So we know the basic composition of a viral structure. Now if you think